So valine is the third amino acid that we're going to talk about in our 20 days of amino acids. So amino acids are protein letters. There are 20 different common protein ones we're going to talk about, and today up is valine. So like all amino acids, it has a generic backbone part, this amino group and a carboxyl group. These can link together with other amino acids and to form chains through peptide bonds, and then you link a bunch of them together, that folds up and you get a protein. But in addition to that generic part that lets them link up, these each have unique side chains or R groups, and these have different properties, and some are big, some are small, some are charged, some are not charged, some are like water, some don't like water, or water doesn't like them. So all of these different properties, and so because proteins have different combinations of amino acids, and these amino acids have these unique parts with these different properties, the proteins are going to have different properties. And so by understanding the different properties of these different unique parts, these side chains or R groups, we can better understand how proteins get their shape and their function and all this cool stuff. So let's talk about valine. So valine, its side group is this like CH2, CH3, CH3. So basically you add on 3 carbons, then you add on hydrogens to get your filler space. And so you can remember valine is like a V, so it's like a V shaped. And it's 1 letter abbreviation is a V, 3 letters is VAL, so val. And it is short and squat, and it's one of what we call a branch chain amino acid, because this is branching out when you're, and because this branching is really close to the like alpha carbon, so to the like the backbone carbon it's attached to, it's gonna make this kind of like hard to, it's gonna not be wanting to be in like helixes and stuff, because it's hard to move because there's all like this bulk if you try to move around it, and it's gonna bang into stuff. So valine is super valuable. In fact, it's essential. So you can remember valuable, valine, essential. But being essential doesn't mean that it's more important than the other amino acids. It just means that unlike some of the amino acids, our bodies can't make it ourselves. We have to get it through our food. And so it is super valuable for a ton of different reasons, but it's not valuable when it's places it shouldn't be, like on the surface of hemoglobin, which is the cause of sickle cell anemia. So basically valine is what we call hydrophobic, so we'll get more into all of this, but basically water doesn't like to hang out with the valine, and so in sickle cell anemia you have hemoglobin, which is this protein that binds to oxygen and carries it throughout the blood. In sickle cell anemia, what happens is this amino acid that water doesn't like to hang out with it, and so normally it's found like buried in the center of proteins, away from the water. And normally on the surface of this hemoglobin, at this one spot, glutamate there, which is charged, and so water likes it and water's cool with it. But then you swap a valine in there, and now water's not cool with it. Like that valine should not be on the surface of a protein. So then if there's like a hydrophobic pocket, so somewhere else where water doesn't really like to hang with, then those parts can join together. The hydrophobic parts come together, minimizing their contact with the water and the water's contact with them. And this can lead to the polymerization, or like the linking up of hemoglobin molecules into these fibers. This happens mostly in the deoxygenated state of hemoglobin. So hemoglobin has kind of different shapes when it's bound or not bound to oxygen. And when it's in the state that's not bound to oxygen, it's really more prone to this linking up, this like fibrillization or polymerization. And so when the hemoglobin's going through the bloodstream, so it's like picking up oxygen, giving oxygen, picking up oxygen, giving oxygen, picking up oxygen, giving oxygen. And you can imagine that if it's every time it's in that like once it's given up oxygen, if then it's like linking up, now you're going to be totally stressing out these cells if you're like polymerizing, I'm polymerizing, and then when it's polymerized, then it can make these cells all like funny shaped, like sickled, and then that can cause blood vessels. So there's a lot of problems, and you can cause so there are different types of sickle cell disease and like beta thalassemias and stuff which are related. But sickle cell anemia specifically involves this specific glutamate to a valine mutation. But there are other mutations that don't involve a valine, and there are other mutations that are more conservative, so they're less like drastic, like charged to nonpolar type of things. And so those can have like more minor effects and various things. So more on that in the sickle cell post. So more about all this later. I just wanted to give you a brief intro to like hook you in, because I know there's different things that will get different people synced in. Maybe you care about the polarity, maybe you care about the sickle cell. No matter, maybe you care about the history of its discovery. Cool stuff all around. So let's talk about Valine. You know, I'm not really one to care about looks, but I do care about the looks of chemicals, and so this is what Valine looks like. It's kind of like this white, flaky, kind of shiny. I'm trying to think of what it reminds me of. Maybe like fake snow in a bit. I don't know, but it's like flakier than the other, which were more granular. But again, I don't know if this is just a processing thing or anything, but just since you probably don't really get a look at Valine very much, there you go. So if you need a refresher on amino acids, peptides, proteins, all that stuff, check out the post from a couple days ago, and we're going to just talk mostly about Valine today. So this is the third one we're going to talk about. So we talked about glycine, its side chain, which is the hydrogen, then alanine, that had this methyl group, and now we have valine. And so we see we have these two methyl groups and this carbon with just a single hydrogen. And because remember, carbon like forms up to like form 4 bonds, and so if you don't have a bond to something more like fun, then the hydrogen has to take its place. But when you have something like valine, we see that it just has a hydrocarbon chain. So it just has like carbons and hydrogens. And when you have carbons and hydrogens, that's kind of just like blah in terms of functionality wise, but it does add like bulk and structure, and does have consequences. And because it's so like kind of blah, that water doesn't really want to hang out with it because it doesn't offer anything for water. So it has this hydrophobic property, and this is important for helping proteins get their folding, because those hydrophobic parts, we have this thing called the hydrophobic effect. So basically the reason why these hydrocarbons are so like blah is because they're non-polar. So they don't have any like separation of charge. So polarity is where you have like separation of like partial charge separation in a molecule. So even if a molecule is neutral overall, it can have like partially charged parts. And this is because the atoms themselves, so like the individual carbons, hydrogens, oxygens, all of these different things, they're made up of smaller parts, protons, electrons, and neutrons. And so the protons are positively charged, and they're kind of like glued together by these neutral neutrons, and they're like stuck in the center of the atom in the nucleus. And the number of protons defines the element. So whether it's an oxygen or carbon or whatever, they differ in their number of protons. The electrons are negatively charged, and they kind of whiz around that central nucleus. They're equal and oppositely charged to the protons. So if you have an neutral molecule, you'll have an equal number of protons and electrons, but you can also have an imbalance, and that gives you a full charge. But even if you don't have a full charge, because these electrons are like whizzing about in these clouds, and atoms like can share pairs of electrons to form bonds. That's how we get these like strong covalent bonds. So like the links between these different atoms. Those are covalent bonds that involve basically the merging part of their electron clouds. When they're sharing their electrons with the other atoms, though, to form these bonds, sometimes the atoms don't share fairly. So remember, the protons are kind of like stuck in place, and they're the pulling power. They have that positive pulling power from the protons. They're trying to rein in these electrons that are whizzing all around. And so when you have shared electrons, sometimes one of the partners is better at pulling on those electrons that they're supposed to be sharing. And this leads that part to be partly negative and the other part partly positive. So these ones that are really good at pulling, we call these electronegative. And so oxygen's really good at that. And so it's going to pull the electrons and make it partly negative. These hydrogens are going to have electrons pulled from them, making them partly positive because they still have that proton, remember? So this gives you this partial charge separation that we call polarity. So because water has this polarity, it has these partially charged parts. Now these partial positive parts on one water will be attracted to the partially negative parts on another water. And you can see you can get these water networks. They'll also let in other things into their network if they have something to offer. So if there's something else that's polar or something else that has like a full charge or
then the water, we call those things hydrophilic because the water can hang out with them. And so the water is going to let them into their network. Some atoms, however, share, if they have like similar electronegativity, so they have different amounts of like pulling power, when they share, then they're going to share fairly. And so you have something that's nonpolar. And this is what happens when we have hydrocarbons. Like, so when we just have hydrogen and carbon, they share pretty fairly. So you're not going to have this polarity, and then you're not going to have any like charge. And so the water is not going to want to hang out with it. But the water is not going to want to hang out with it. The water is going to want to hang out with other waters. And so when you put a hydrophobic thing into water, the water is going to try to maximize its water-water contacts and minimize its water hydrophobic thing contacts. And so it's going to kind of gang up around those hydrophobic things and kind of like cinch them together. And so if you have these hydrophobic things like coming from hydrophobic groups on an amino acid, and then it's part of the protein chain, this is going to influence how the protein folds so that those hydrophobic things are stuck like in the central part of the protein away from the water. And so you can see that valine is classified as very hydrophobic. So what happens then if valine is on the surface of a protein? Well, this is what happens with sickle cell anemia. And so basically hemoglobin is this protein that carries oxygen through your blood. And it's made up of these like 4 separately made protein chains that work together. And this beta globin chain, people can have diseases where they have mutations in this beta globin that can cause this hemoglobin to like malfunction. And there are different mutations that can cause different problems, but a very common one is sickle cell anemia, which is caused by like this specific mutation at this site. And basically this mutation, it causes a mutation. This mutation causes a change from a glutamate. So that's where the E was from. It's glutamates, the abbreviation is E. We'll talk about glutamate more later, but you can see it's just negatively charged. And now we have, in the sickle cell version, you have a valine, and we talked about valine being really hydrophobic. So what happens then is that if you have this hydrophobic amino acid on the surface of the protein, instead of like hidden away from the water, now what's going to happen is you have it makes it kind of like sticky. So if there's another hemoglobin molecule that has a hydrophobic patch, so something else that's hydrophobic. So remember that if you have hydrophobic things, the hydrophobic effect is kind of going to like force them together. So that the water doesn't have to like get as ordered around it. So water likes being really disordered. And so if you have something, like the more things that water has to kind of like cinch around, the more you have to like use up water to kind of like control those things it doesn't like. And so forcing together the hydrophobic things gives you this like entropy benefit where you're like kind of free the water to be more free. So what happens so especially in the deoxygenated state, so basically hemoglobin, it takes on like different shapes. So when it's oxygenated and deoxygenated, so when it's oxygenated, it's in this like R form, and deoxygenated, it's in this like T form. And what happens is that in this T form, so the deoxygenated form, if the sickle version can like link up to form these long fibers, and these like long helical structures that can cause the red blood cells to get misshapen and clog blood vessels. And so basically this every time, since it's happening in the deoxygenated state, and the hemoglobin is constantly, as it's like getting circulated, it's going from oxygenated to deoxygenated to oxygenated to deoxygenated. And so it's going sickle, unsickle, or like the cells are like sickling and unsickling, and as the fibers are forming or unforming. And once it does that like 40 to 50 times, then it's basically permanently stuck in this sickled form. And at the sickled form, then it can clog these blood vessels. And so there are different like therapies and stuff, some gene editing things in the work, more on that in the sickle cell post. But yeah, so that's a consequence of like having that hydrophobic residue out on the surface. So valine is coded for by the codons that start with GU. So this means that when the ribosome is traveling along the messenger RNA, every time it comes to 3-letter sequence that starts with GU. The tRNA loaded with the valine is going to come, and the ribosome is going to put that valine, add that growing chain onto the valine, shift things over and keep going. And but yeah, so those are the codons. So you will find it in this corner of the table. So valine, it's a branched chain amino acid. So it's an aliphatic, which means that it has it's a hydrocarbon that is not a ring. So we'll talk about these like aromatic. These ones are aliphatic. So they have these hydrocarbon chains that are not rings. And you can see that valine, leucine, and isoleucine, they're all branched chain. So alanine is not, because it doesn't have a branch in its chain, but these have a branch in their chain. And when you have a valine, you can see that this branch is coming like right off of the alpha carbon. And this is going to restrict the motion. So we talked about how like motion is already restricted based on like the peptide bond having this partial double bond character. This resonance stabilization is making it so you can't rotate around this bond, giving you kind of like a chain of planes that you can rotate around. But the amount of rotation that you can do in these different places is then going to be limited by the side chain that's there. And when you have something small like glycine, we saw that it could be really flexible. But when you have something that's like bulky, like branched, like close to the like that has that branching right near where you're supposed to be, where you're trying to turn, that's going to restrict its motion. So it's not going to be found in like helices and stuff very much. And it's definitely not loosey-goosey like glycine. The branching also means that it is going to be harder to break down. So the branched chain amino acids, they have this like special breakdown pathway, involves a couple of enzyme complexes. We'll talk about it in a later post. But basically there's this branched chain aminotransferase. So basically the first step is they're going to remove this amino group, and then they are going to convert it, break it down further. And so mutations in the second complex can lead to this thing called maple syrup urine disease. So we'll talk about it in a later post. But basically with valine, what you're going to do is in that first step, when you remove the amino group, you're going to pass it off to alpha ketoglutarate to make glutamate. So we talked about this in our post yesterday on alanine. So alpha ketoglutarate is this product of like cellular respiration. So when your cells are like breaking down like sugar and stuff to form energy, there's this tricarboxylic acid pathway. Yeah, so you might heard of glycolysis. Start with like glucose, a blood sugar, you break that down, you get pyruvate, that enters the citric acid cycle. And the citric acid cycle is where you're going to be finding the alpha ketoglutarate, also called 2-oxoglutarate, and this cycle is also called like the Krebs cycle or the citric acid cycle. But the basic thing is that there's this intermediate in it called alpha ketoglutarate, and it can, if you give it an amino group, then you form the amino acid glutamate. And then there are pathways and stuff that can use this glutamate, and then safely get rid of the amine group. And so, yeah, so like the first step in amino acid breakdown is removing that amino group safely. And so this is how you do it, by transferring it to glutamate. Once you do that, now you have an alpha keto acid. And so a ketone is where you have an oxygen double bonded to a carbon, and then that carbon has carbons on either side. So this is an alpha keto acid. In this case, it's alpha keto isovaleric acid. And then that's going to go through that second complex and turn it into this isobutyryl CoA. More steps, you'll get succinyl CoA. And then that succinyl CoA can enter the cycle. So when it enters the cycle, now we can go and you can actually make glucose from it. So we call it glucogenic. So we call valine glucogenic because you can make sugar directly from it. let's see, break it down. So now for the discovery stuff. So from my blog, so basically the story begins in 1856. There's this chemist named von Bessenes. And so he was trying to figure out what was in glands. So like he took a pancreas, minced it up, and extracted stuff. And so he found, he was looking for specific amino acids and he found them. So he found leucine and tyrosine. But he also found something that behaved a lot like leucine, but it was less soluble in boiling alcohol. So leucine, remember, it's going to have additional carbon, so it's got like a longer chain before the branch. So you can expect it to act pretty similar, but also there would be key differences. And so he noticed that it wasn't really leucine, it was slightly off. So it has these different properties. And so then he figured out the empirical formula. So basically he figured out the number of, like the ratio of the different atoms in it. So he knew how many atoms there were compared to one another, but not how they were connected. And so he knew, okay, this isn't leucine. It's got fewer of these like carbons and hydrogens, but I don't know what it is. I don't know how those atoms are connected. Then in the 1880s, other groups found it in albumin and sprouts of Lupinus luteus and Vicia sativa. So it was found in a variety of places, but they still didn't try to figure out like how those atoms were linked together. Other scientists did, but they had a hard time figuring out how they were linked together because there's a lot of possibilities. So most of the first half a year of research on what we now call valine, it was different chemists basically trying to synthesize different possible combinations and then comparing them to how the real thing acted. But they were having problems because their synthesis products contained a mix of stereoisomers. And so remember we talked about how stereoisometry is kind of like handedness, where you can have things have the same like connectedness, but like different orientations in space. So kind of like your right hand and your left hand can be like mirror images, but you can't like superimpose them. So basically in our bodies, we all use the L form, but when you synthesize it in a lab, you get this mix. And so when they were doing this stuff in the lab, they were getting this mix, and so it wasn't acting quite like just the L alone. But then in 1906, the new separation techniques came along, and then he was able to separate those stereoisomers, so separate the L and the D, and nail down the correct linkage. And so this was Fisher, and so then from thenceforth, the name valine would refer to alpha amino isovaleric acid. And so this Fisher, he went on to do a lot more work on amino acids and synthesizing peptides. And he did a lot of good stuff and he did some bad stuff. He tasted his chemicals, which remember, you don't taste things in the lab. But anyway, he did this and he found that the L and D stereoisomers actually taste different. So the way that you can tell them apart lab-wise is that they rotate light differently. But D-valine is apparently bittersweet and L-valine is all the way sweet. Yeah, so hope that helped you get a better appreciation of the value of valine.